Thank you, Jim, for the kind introduction. It's a pleasure to be here. Thank you for having me, for inviting me. I don't work on aging. I was invited by an institute on aging, but the closest thing I do to aging is a study of circadian rhythms. What I'm really interested in, what our lab is really focused on, is understanding intelligence in brains and machines. That's what is really the focus of our lab. In doing that, sometimes we try to develop complex informatic systems for understanding biological data, in particular omic data in this case, and we apply it to different problems. And the one I'm going to describe to you today is a study of circadian rhythms at the molecular level. If you saw the announcement for my talk, I had a more complicated title because I thought that I would talk about circadian rhythm for half of the talk, and then the second half I would tell you a little bit about deep learning, which is the bleeding edge of machine learning today. But in preparing the talk, I realized it's too much material. So I'm going to focus on circadian rhythms. I still have a 15-minute presentation on deep learning at the end. If we have time, if you have no questions, I could do that. But most likely, we are going to stick to circadian rhythms for this talk.
Pierre?
Yes?
I think you're double mic'd.
Oh.
Push that away.
So. Nevertheless, I'm going to give you a little bit of background on the big picture about computing, and then some background about circadian rhythms. Probably most of you are familiar, very familiar with circadian rhythms. And then the meat of the talk is the development of methods for understanding these large amounts of omic data, different types of omic data, integrate all that information, and to come up with what I think is a new framework for thinking about circadian rhythms at the molecular level. So imagine that an alien, this is a picture of an alien from a distant galaxy. An alien is sent to planet Earth, and his mission is to report on the state of computing and intelligence on planet Earth to his chief on this distant galaxy. What is the first thing he would have to say? The first thing you would have to say is that on planet Earth, there is really two types of computing. There is silicon-based computing, your computers, your cell phones, etc. And then, of course, carbon-based computing, which is what all living systems are implementing. And you and I know that carbon-based computing trumps silicon-based computing, of course. And these two ways of computing are completely different. You can analyze them in terms of time scales, spatial scales, et cetera, energy, architecture, and you can see that they are enormously different. For instance, energy-wise, the brain consumes on the order of 20 watts, a couple of light bulbs. The IBM Watson supercomputer that was built just for the game of Jeopardy initially, this is a supercomputer that has human or even superhuman performance in the game of Jeopardy, consumes 85,000 watts, so 3 to 4 orders of magnitude more. Incredible difference in energy efficiency. And the architecture is very different. I think the main difference is that in your computer, memory and processing are separate, completely separate. In the brain, as far as we can tell, they are completely intermingled. So the architecture is completely different. And of course, the hope is to gain some insights from the architecture of the brain to develop better computing devices over time. There are a couple of similarities I want to point out to you, however, is that both systems have converged on the idea of using tiny electrical signals to communicate between processing units. This is perhaps not surprising. You cannot imagine having a system that uses mechanical forces or sound to transmit information between its components. And the other things they have both converged on, as far as I can tell, is learning, the idea of using learning. Hence, machine learning is very important today within AI, is probably the most important sub-areas of AI today is machine learning. And deep learning, as I mentioned, is really the bleeding edge of machine learning today. And that's why I wanted to tell you a little bit about that, and maybe we'll do that at the end. And of course, biological systems have circadian rhythms. which are a very strange thing, because you may think that your life is ruled by your spouse or by your dean or by Obama, but in reality, it's really ruled by your circadian rhythms. Every day, you go through a very mechanical routine in a very robotic fashion, I should say, which is dictated by circadian rhythms. And if you rebel, as you know, you become a rock star and you die, basically. So you can't rebel. And the reason we tolerate circadian rhythm is that, on the other hand, they are very gentle. It's a very gentle boss. You want to have a lunch at 1 instead of 12, no problem. You don't even ask for permission, right? You want to watch a movie at night, same thing. You don't ask for permission. The only time you notice them is if you travel and you have jet lag, and you think that after a few days the jet lag goes away, which is something that remains to be shown at the molecular level. I don't believe it goes away. But still, it's a very strange phenomena where you have this very rigid rhythm in some way, which is also at the same time very plastic and flexible. If you want to understand circadian rhythms at the molecular level, of course, what you have to do is to try to reverse engineer the circuits, because as it is in silicon, the circuits, of course, are designed by us, but in biology, they have been evolved, which is a very different process. We have to use reverse engineering methods to try to understand how circadian rhythms are implemented at the molecular level, and so that's the kind of techniques I will be describing in the talk. So circadian rhythms, you are probably all very familiar with that. They are very conserved. They are found in fungi, cyanobacteria, in plants, of course, in mammals. So throughout all living systems have some kind of circadian rhythms. This has been known for quite a while. And it is known that there is an endogenous component to circadian rhythms. They persist at least for a few days in total darkness. It's true in plants, for instance. It is known that disruption of circadian rhythms can lead to all kinds of problems, depression, obesity. There are links to aging, sleep disorders, etc. So they are very important for health, of course. And the center, in some way, of our circadian rhythms is in the brain, in the hypothalamus. There is a nucleus called the suprachiasmatic nucleus, which has about 30,000 neurons, which are highly rhythmic, which are believed to be the center of the circadian rhythms. They receive direct input from the retina, for instance, so the light is called the Zeitgeber in the field. Zeit means time in German, Geber, time giver. And there are other Zeitgebers, food is Zeitgebers, etc. But when you look at things more closely, you find circadian rhythms everywhere. It's not just in the hypothalamus. Every cell in your body is actually oscillating in a circadian way. This has been shown many times. For instance, using DNA microarrays or RNA-seq, you can show that there are genes that are oscillating, transcripts that are oscillating on a 24-hour basis in every cell of your body. And the core machinery that is believed to be responsible for that it's quite well understood. It's about a dozen genes. The main players are clock and BMAL. These two proteins can dimerize. They bind to DNA, in particular to regions called E-boxes. I'll show you how E-boxes look like. These transcription factors bind to these E-boxes. They activate the transcription of a number of genes, in particular genes in the CRY and PER family. So there is CRY1, CRY2, PER1, PER2, PER3, etc. These proteins go about their business. They get phosphorylated, and finally they come back. They form a complex that is not well understood that binds and inhibits clock and BMAL. But that is really the core, what is called the core clock at the molecular level, which operates in any one of your cells. On the other hand, when you do DNA microarray experiments, you're going to find a much larger number of genes that are oscillating in a circadian manner. And that's sort of the kind of problem and theory that I'm going to describe to you today. So what we need to do is to build methods that allow you to sort of automatically derive this little diagram that biologists like to draw when they are explaining some biology, where you have receptors, you have signaling pathways, kinases, etc. You have transcription factors. And in the case of the little machinery that I just showed you, this is a classical transcription translation inhibitory feedback loop, which has this 24-hour periodicity. But of course, in the cells, you have many loops, and it looks very complicated. The question is, can we use computers today to build these circuits and to try to make sense out of them in a somewhat automated fashion? And I'm going to show you how I think we can do that to some extent. And the question is, where should you start? You know, some people like to start from gene expression, the geodatabase that has lots of microarray data. You could start from epigenetic, et cetera. I like to start from the genome because the genome is a well-defined type of information. You have the DNA sequence of a given, say, mouse species. That is a sort of well-defined object. You have the entire genome available to you, whereas if you have microarrays, it's much more noisy. You have continuous values, et cetera. So we like to anchor things in the genome. That's the first thing you want to do. And today, of course, we have genomes of all kinds of species, even different strains of mice, for instance, that are available to us. And the second thing, so you start from the DNA. And the second thing, again, at a very high level, you look at the central dogma of molecular biology, which says that information flows from DNA to RNA to proteins. Well, the most central things about this dogma is that it has to be wrong, right? You cannot have something that works in feed-forward mode and produces life. You need to have feedback. So the first thing you have to do next, once you have DNA, is to put feedback in this loop. And where is the main feedback? The main feedback is really from proteins back to DNA. In your genome, you have about 20,000 genes. About 10% of them, 2,000 of them, are transcription factors. That is, these are proteins that go back to the DNA, bind to the DNA in complex ways, and control the expressions of genes. So the second main step that I want to do, once I have the genomes, is to study and establish a map of the feedback from proteins to DNA. Now, how do you do that? Well, the first thing you do to analyze this feedback, you need to map all the places along the genome where the transcription factors are binding to. So to create a genome-wide map of all the places where regulation might occur. So you go into the databases of transcription factors that are available today. The two main ones are TransFac, which unfortunately is commercial. There is JustPar, which is freely available. It's a little bit smaller. But anyway, these databases contain a list of transcription factors. In these databases, you can find about, a little bit over 1,000 transcription factors today. So it means that for half of them, the data is missing. You have to be aware of what's missing. But you have about 1,000 transcription factors in this database. And for each transcription factor, you have the matrix or the signature, the motif, the DNA motif that this transcription factor likes to bind to, which has been obtained by biologists through long experiments, decades of experiments, maybe ChIP-seq experiment today. So for instance, P53, a very important transcription factor involved in apoptosis, in cancers, and all kinds of other biology, has a binding matrix that looks something like this. It likes to see a G in this position, it likes to see a C in this position, et cetera, et cetera. So of course, this is converted to probabilities. We have those probabilities from this matrix. The next thing you do, of course, is to scan the genomes with this thing and find all the places in the genome where this motif is found. Okay? Now, P53 seems to have a long motif. It's fairly discriminative. You're going to find a relatively small number of positions. But a lot of transcription factors may have, as far as we know, a much shorter motif. It may be degenerate, et cetera. So in many cases, you're going to find a lot of hits. And probably a lot of those hits are not functional, although we don't know for sure, right? So the question is, how do you filter all these hits that you get after scanning the genome with a few thousand matrices and scanning all the genomes. By the way, you can do this for human, for mouse, for yeast, C. elegans, etc., all the model organisms. The basic filter, the main filters we apply is comparative genomic. So this is an example of what happens if you look at rat, mouse, dog, human. You align the genomes. There is a genes that start here. Doesn't matter what gene it is. And I'm looking at the promoter region upstream of the transcription start site. And you can see there's a lot of variability, etc. There is no particular signal, except in this position where you see almost perfect conservation about this in these four organisms. That, of course, tells you that this little piece of DNA must have a function. Otherwise, evolution would have tinkered with it. And indeed, this is the binding site for this transcription factor here. So what we're going to do is to align a lot of genomes and look for hits that are reasonably conserved across multiple species as a filter to clean up our map. So that's an example of a tree we use. All these genomes by now have been sequenced, right? Or this is another little deeper tree, the deeper trees, etc. So there is a question of what tree to use, and I'm not going to do in detail into that question, but that's the tree we're using for what I'm going to show you. And so the next question is, how do you measure, as precisely as you can, evolutionary conservation? So imagine you have genomes that are aligned here, and you think that you see the motif, let's say for P53 here and here, etc., in the genomes. How do you measure the degree of conservation? One idea is to compute what is called the branch length score. So you have your tree, you look at the most recent common ancestor to all the red dots, and then you add the length of all the branches. That's called the branch length score. The more conserved the motif is, the deeper is the tree, the longer the total branches are going to be, right? So that was introduced around 2007 and shown to work reasonably well. But you can improve on that because, of course, you don't get 0 and 1s in these positions. You get a probability score associated with how likely it is that the motif is present at that position, right? So can we leverage the information that is in these probabilities when computing the branch length score? And so you can come up with something we call the Bayesian branch length score, which is a little bit different. And again, on a simple example to show you, suppose you had 4 species with this tree, and you see the motif in this position and this position. This is the most recent common ancestor, so the score would be L3 plus L5 plus L1, right? Now, if you want to do this probabilistically, imagine that you see it in all 4 species with probability 1/2. Again, this is just a toy example to show you how it works. Then you have to look at all possible assignment of the motif to the leaves. These are all possible cases. Each one has a probability of 1/16 because all the probabilities are the same. And in each case, you would get a completely different score. And so you can take the average, the expected value of all these numbers, and you get this, which you see is quite different from what we had in the previous slide. So you can call this the expected branch length or the Bayesian branch length. And so that's what we're going to use. And we have shown that it works a little bit better than the other methods. So now we have, after doing all this work, we take a species, we find all the places where the matrices of the transcription factor hit the genome, and we have a score, which is the probability that the motif matches the DNA at that place. We have a second score, which is the conservation score. And then we can also have a so-called false discovery score, which is obtained by scrambling the columns of the motif and running them against the genome with the scrambled version. That's a way for getting an estimate of the false discovery rate. And how do we assess the quality of the map? Well, there is lots of things you can do. We can test against ground truth. So ground truth is what you get with ChIP-seq or ChIP-chip experiments, where you have a transcription factor and you test it against the entire genome by these experimental methods. They are not perfect, etc., but you can use that as a sort of approximation to the ground truth to see how good these computational methods are. You can, of course, collaborate with biologists, predict binding sites and have them test in the lab. If you're lucky, you can get those kinds of collaborations. You can also analyze where in the genome you're finding these regulatory sites. Ideally, you would like to find them in promoter regions, not too far from transcription starts sites, et cetera, et cetera. So I'll just give you a few examples of these kinds of tests. So this is a transcription factor called NRSE. This is the logo. These are different methods that have been proposed for analyzing this conservation. This is the branch length score. This is the Bayesian version. And these are so-called ROC curves, a perfect curve which are plotting false positive rates against true positive. So a perfect detector would go straight up here and then be flat. And you can see that at least in this example, comparing to experimental data, the BBLS curve gives you the best results. They're not perfect, but it's the best methods. And we have done this now on all kinds of different important transcription factors for which a lot of high throughput data is available. This is CTCF, P53, et cetera, et cetera. I could show you a bunch. You can measure the area under these curves is called the AUC metric. It's a standard metric in these kinds of, to assess the performance of classifiers, of predicting systems. So you can measure the AUC, and you can see that the BBLS scores gives you the best AUC on the order of 80, 81% in many cases, not always, but a pretty good AUC derived by this metric. This is a location analysis. If you plot the histogram, where is the majority of these sites found, you can see indeed that it's quite close, within 20 kb of the transcription start site. There are a lot of transcription factors that are binding the first intron, so it's not a surprise to us that you find things on both sides of the transcription start site. So this is another sign that at least globally, we're getting reasonably good maps of these regulatory sites. This is an experiment that was done in collaboration at UCI with biologists that are working on Drosophila. So this is a transcription factor that has a spacer here, if I remember correctly, of 5 base pairs. So if you increase this to 6, it doesn't work anymore, it doesn't bind. So that's how you can test that the binding site is active or not. And this transcription factor is involved in the development of the eye in the fly. And you can see that when the binding of the transcription factor at the right positions, you get this kind of distribution of receptors in the retina. And this is when the binding site is disrupted, you get something which is much more disordered. And so using this method and searching the fly genomes, we had a prediction of 13 sites. And of those 13, 8 were shown to be functional by this method. It doesn't mean the other 5 are not functional, but by these detection methods, 8 out of 13 were shown to be functional. And so we serve this to the community through a website that you are welcome to visit, which is called Motif Map, which provides these genome-wide maps of regulatory sites. You can choose your transcription factors, you get the logo, you get all the hits in the genomes, you have different score that you can adjust if you want to increase the stringency of conservation, for instance, et cetera. You get the list, you get what's the nearest gene, et cetera, et cetera. So this is a tool that is widely used. You can even download the data. We make all the data available. So that's one core technology that we have developed. And I want to point out to you that just doing this already is the beginning to obtaining circuits, because if I say P53, which is a transcription factor, I can ask what is the list of genes that have a P53 binding sites in their promoter region, for instance, and you get a bunch of genes around, and you can put an edge, so that's already the beginning of drawing those circuits that I described to you. Then you can start superimposing additional information. For instance, there are databases of protein-protein interaction, biobase, PPI interaction. So I can put edges now between these nodes representing an interaction between this protein and this other protein, okay? Of course, you have to be very careful. It's not because you're drawing an edge it doesn't necessarily mean that the interaction is in the biological system or the particular system you are looking on. There is all kinds of other layers. There is mRNA, mRNA to proteins, there is epigenetics, et cetera. So you have to be very careful when you're drawing these graphs about what information you are leaving out and what is the meaning of the edges you are drawing. But you can see that it's the beginning, and it provides a substrate where you can add additional information on top, and I'll show you how we add information about circadian rhythms on top of this diagram. Again, to test the accuracy of these methods, you can take P53, a transcription factor that has been studied by decades for biologists, and you can ask which proteins are controlled by P53. Again, I can search 2 kb upstream of the transcription start site. This is the pathway in the CAG database of pathways, which tells you what is known about P53 from the literature. You can see that with 2 kb, we start hitting quite a bit of them. And if I increase the window, if I go 10K up and downstream, I recover most of the pathway. I can recover, decades of very hard molecular biology just using informatic methods and using this large omic data that is available to us. And this is going both upstream and downstreams, looking at neutron, and then most of the known pathways are recovered. In fact, you can do even more. For instance, again, this is P53. I extracted this from the previous figure. We have a prediction that it hits these 2 proteins here. So that seems to be a false positive if you believe the data in CAG. But we went in the literature, which is another data source. You can search all the abstracts in PubMed for articles that have these things and P53 in them. And indeed, we found them. And we found that, yes, it has been reported in the literature that P53 regulates these proteins. So this is actually, most likely, this is a false negative in CAG. It's not in CAG, but it should be there. But you see, you have to integrate information from different sources. It's this integration work, taking data from the PubMed, from Geo, from GenBank, SwissProt, et cetera, and stitching it together. And you never want to believe something that has only a single data source. You need to have several sources impinging on the same thing in order to really give it some credibility. Another example of a project with a biologist at UCI, Bogi Anderson, who studies skin, wound healing, skin development, etc., is interested in a transcription factor called grainy head. So we take grainy head, we can immediately build a circuit around grainy head, just looking at transcription factors. Which other transcription factors are controlled by grainy head via motif map, or are controlling grainy head in the sense they have a binding site in the promoter of grainy head. These circuits between transcription factors, we believe, are very important because those are relatively fast-acting circuits which are really the center of the regulatory machine of the cell. So we get a circuit like this, and I'm going to zoom in into grainy head, which is here. And so we predicted immediately that grainy head should be controlling this other transcription factor, including oval-1 and oval-2. At the time, this was not known, and Bogi has worked on this very hard, and by now, yes, it has been shown that this is exactly the case. So you can generate interesting hypotheses that are important for the biologists, for the biology, and that can be tested in the lab relatively easily. So I go back to the Watson computer, because the Watson computer built by IBM is doing exactly this for Jeopardy, right? It takes data from the New York Times, from Wikipedia, from the Encyclopedia Britannica, takes all this data, integrates it in some way, and uses that to play the game of Jeopardy. So that's basically the kinds of things we're doing here. We're taking all the omic data we can find in all kinds of different databases, and the question is, how do you put it together? And I try to give you a flavor of that. In fact, we call our system, this integration, CRICK. This is a pun on the name. Watson is T.J. Watson, which is the founder of IBM. This is Jim Watson and CRICK, right? So we call our integration system CRICK. This is not publicly available just because it's too big and messy. I wouldn't know how to serve it to the group. But just to give you a sense of all the different databases that you can overlay, for instance, drug you're interested in drug discovery and drugs, you can download the list of all FDA-approved drugs with their structures, and when it is known which is their target protein, and on the graphs that I was describing, you can put the small molecule, the drug, that is attached to the corresponding protein, and try to understand, for instance, side effects or any other things. So it's a fairly powerful method for integrating all kinds of different data, and I won't go into all the details. So now let me move to circadian rhythms, and this is a collaboration with Paolo Sassone Corsi, who has a big circadian lab at UCI. He likes to have his picture taken always with a clock in the background, or you can see here with this watch, working on circadian rhythms. And I'm going to tell you about a different system that we have developed, which is called Circadiomics, which uses the previous one, motif map and CRICK, just for the study of circadian rhythms at the molecular level. And the first experiment we did with him produced this kind of circuitry. So you can see immediately that one of the problems is how do you make sense of this big circuit? It's nice that they are generated automatically, but inside this big mess, what are you going to focus on? How do you rank this is a bunch of hypotheses that the system is spitting out. It's saying that this node is controlling this node, et cetera, right? How do you focus on what is the most highest priority of interest to the biologists? You almost have to build another Watson system to analyze the output. But fortunately, if you work closely with biologists, they already have narrowed things down. Maybe they are interested in the Thor pathway or P53 pathway or clock and BMAL in the case of circadian rhythms. So you can focus on particular isolated regions. And so what Circadiomic does is take the circuitry that we have shown to you, but superimposes circadian data. So today you can do circadian transcriptomics. So you look at, you do microarrays or RNA-seq in a circadian fashion. You take mice, and then every 4 hours you extract mRNAs and look at the profile. And in this first study we did a couple of years ago, was the first study to do it for the metabolome. So you can do high throughput mass spectrometry. This was done through a company called Metabolome, where you can measure the level of the metabolites in a circadian fashion. Ideally every hour, but that's too expensive, so you do it every 4 hours and things like that. But that's what we did in this experiment. And so when you overlay the information, what you see here, so we are representing in this graph the different levels of information. This is the promoter of this protein. This is so what you see in each box, first of all, you have day and night. In each box is a different molecular species. And what you see is the profile, the circadian profile that is empirically measured, right? So you can see that this protein, UPP2, is an enzyme that controls these two metabolites, uridine and uracil. And this is the oscillatory, this protein is known to oscillate. At least the mRNA oscillates. We don't always have the levels of the protein because that's more difficult to measure. You need to do mass spectrometry. It's not high throughput, et cetera. But anyway, the mRNA, it's easy to measure and is oscillatory. Presumably, the level of the protein is also, of this enzyme is oscillatory. And it controls these two metabolites, which we were able to measure in a circadian way. And what you see here is the profile in a knockout where the oscillation is completely suppressed. Whereas in the wild type, in this case, you can see a nice circadian oscillation. And you can ask about how is it that this protein has this circadian profile? Well, you look at its promoter. You look at all the proteins that are binding to this promoter, all the transcription factors. We have the mRNA profile of all the transcription factor. And guess what? Clock and BMAL, which is also called RNTL, these two proteins are the two proteins that I told you are in the center of the molecular clock. They are oscillating. It's known they are oscillating on a 24-hour basis. So you can make a story, make a story of what is going on in this picture. And this is just, a small area of that of the large graph that I've shown you. So we can zoom in and look at specific metabolites. The Sassone Corsi lab is particularly interested in the effect of diet on circadian rhythms. So we have had mice on high-fat diet. We have done ketogenic diet, so-called Dr. Atkins diet. We haven't done, but of course it's on the to-do list, to do calorie restriction diet and to see how these diets are going to change, if anything, are going to change in these circadian rhythms that you have at the molecular level in these circuits. So that's the big picture of what we're doing. And we serve, again, all this through a website called Circadiomics. You can go there. You can enter the tissue type, liver, we have gut, we're going to have brain, etc. You can enter either the name of the gene or the metabolite. And if we have data on it, we'll display to you what is the circadian profile that we have. You can click on View Network, and you will get the corresponding network of that area. So that's just infrastructure building. So now what I want to show you is once you've built this infrastructure and then you applied to these circadian experiments, some of which are ongoing, what is the sort of new picture of circadian rhythms that I'm trying to get to? So what I've done here is aggregated results from maybe 15 such experiments. These are all transcriptomic experiments, so measuring the level of mRNAs by microarrays or RNA-seq in all kinds of different conditions. And this is also across different tissues. Some is liver, some is gut, some of these are knockouts, some are different diets, etc. And the big pictures that you're getting is that in any tissue, you take any cell, about 10% of the genome is going to be found to have oscillations. So way beyond the 12 genes or so which are in the central clock. Okay? About 2,000 genes plus or minus 5% are going to oscillate in a given cell in your body. Okay? That's the first interesting finding, and that was known. But the most interesting thing is that when you look at the overlap between these different experiments, which is what this matrix is described to you, you find very little overlap. So if you look in your gut and you look in your liver and you look at oscillations, you're going to have 2,000 genes oscillating in the liver, 2,000 genes oscillating in the gut, but the overlap between the two is going to be quite small. It's going to be in the range of 200. Okay? And of course, inside of that, you're going to find the clock that has 12 genes. So very little overlap across these different systems, which means that if you aggregate the data together, you find that almost any gene can oscillate on a 24-hour basis in some condition, in some system, some tissue, under some diet. Almost any gene in the genome, we predict, is capable of oscillating. If you aggregate these things, you're going to find like 12,000 genes. It's not the entire genome, but it's not all possible conditions either, right? So it may not be every possible gene, but a very large fraction, 75%, et cetera, is capable of circadian oscillation in at least some scenario. So the real question is, how is this even possible? Is this true, and is it possible? Yes?
The expressed genome in central nervous system is usually about twice that of any other tissue. The same rules apply there?
Yes, we believe so. I mean, nervous system, again, Jim is going to tell you, what do you mean by nervous system, right? If you do this, I believe if you do this in cerebellum, if I do in cortex, you do it in the hypothalamus, you're going to find different sets of genes that oscillate. And with different diets, it's going to be even different genes. That's, again, the central clock. This is the same exercise done for metabolites. So there is maybe 4,000 metabolites. You cannot measure all of them, or it's very expensive. But out of 400 or so that were measured across these different experiments, we find a large fraction, again, that is oscillating. So the same is true for metabolites. It's true for transcript, and I believe it's true for proteins, although it hasn't been measured because of the problem of high-throughput proteomic being very expensive and not really doable today. But I claim any molecular species in the cell is going to be, essentially any of them, is going to be capable of oscillating on a 24-hour basis. These are some examples of perturbations. So you can have a knockout mice. This is an example on the experiment. Again, 400 genes in the intersection, 1,000 here, 238. A diet change, normal chow versus high fat. These are measured in the liver. This is the kind of numbers you get. Even changing the strain, when you say wild type, it means absolutely nothing. There is all kinds of different mice, wild type, and they may be living in different conditions. So you take two wild types, you're going to see different, this is again liver, you're going to find very different oscillatory signatures in their RNA-seq or DNA microarrays. So you have a system that is oscillating, you perturb it, you expect to see changes in the existing oscillations, right? You expect maybe some of the amplitudes will increase, some of the amplitude will go down, maybe even be suppressed, so some oscillation will disappear. You expect to have changes in phase, advanced phases being advanced, phases being delayed. But here we see even more than that. We see completely new oscillations. When you go from one diet to another diet, you have things that were not oscillating that suddenly start oscillating. So again, how is this possible? That you have so many oscillations and so much variability. Is this something we can try to build a framework to understand that? And so that's what I'm going to finish with. These are just examples of the phase advance, phase delay, etc. How is this possible? So again, core clock has a dozen genes. Any tissue condition, you're going to find 10% plus or minus 5% of transcripts or metabolites are oscillating. Overlaps are very small. And when you perturb this oscillation, you see changes in amplitude, changes in phase, but you also see massive new oscillations. Now, if you think about this, a single molecular species does not oscillate by itself. You cannot have a metabolite oscillating just by itself. It's impossible. You need to have a feedback loop around that molecular species in order for it to oscillate. So what is really, when we say oscillations, what we really mean are feedback loops inside the cell which are oscillating in a circadian manner. That's the first thing to observe. We have gazillions of such feedback loops, as you have seen from those diagrams and as you know from biology. So there is a lot, the potential is there. There's lots of loops. So that's great, a lot of potential oscillators. But now, why would they oscillate at 24 hours? Why don't they oscillate at 7 hours or 43 hours? Why do they oscillate at 24 hours, or at least have a propensity for oscillating at 24 hours? Well, the best answer I can come with, so you have to understand that physical objects have intrinsic frequencies at which they oscillate. The classical example is this bridge that was built, I think, near Seattle 100 years ago or a little bit less, which the engineers didn't realize that it had intrinsic frequencies that were in resonance with the winds. And so the bridge started oscillating, and of course, with a high amplitude and broke. So physical objects have intrinsic frequencies at which they oscillate. So these loops in the cell must have intrinsic frequencies at which they oscillate. And one of them has to be 24 hours. How is that possible? Well, I believe it's because of evolution. You can ask yourself, how many times have we gone through a day-night cycle since the origin of life? Easy. 3.5 billion years times 365 days. Easy calculation. My kids can do it. It turns out it's 1.3 trillion times. 1.3 tera. I could put tera with 2 R's as a joke, but 1.3 trillion times. That's a gigantic astronomical number. And of course, you know that, for biological systems, day and night are really extremely different, right? The winds, the pressure, salinity, the tides, everything, the predator, it's very, very different out there. And so it's not surprising, at least to me, that biology has paid a lot of attention to this relentless day-night cycle for 1.3 tera revolutions, and that this sort of relentless frequency, little by little, has built itself in maybe not all, but a large number of these feedback loop that you have inside the cell. In fact, the number is even bigger because the Earth is rotating, the period is becoming larger and larger due to some kind of friction from the moon. There are tides, it's well understood, it's called the tidal acceleration. It's little by little, over evolutionary times, the day-night cycle is getting longer and longer. So 700 million years ago, the period was more like 20 hours. Okay, so the number is more like 2 trillion times that a biological system have gone through a night to day shift and vice versa. Yes?
Curious.
Do you think, so your argument is about the day and night cycle. So would you predict that you might see a different oscillation in, say, the poles, in which the day-night cycle is very different from that of the equator?
Are there a lot of living systems living exactly at the pole?
I don't.
know. I'm just curious.
I would predict that if you are on a different planet where there is life and the planet has a completely different period, I would predict that the circuitry, assuming that it has a carbon-based life, I would assume that the oscillation would be at a different frequency. Absolutely, yes. That would make a lot of sense, right? So that's why in the cell you have lots of oscillators, and they have the potential, many of them, I believe, because of this phenomenon, have an evolution, have the potential for oscillating at 24 hours. So now what happens? All these oscillators are, many of them are coupled to each other in many different ways, and we don't have enough information to really model this coupling. It's a very complex story, but you can see all kinds of mechanisms by which these oscillators are coupling. For instance, I told you about clock and BMAL, these transcription factors which are in the core clock. They bind to a lot of promoters, not just CRY and PER type of genes. In fact, so they bind to E-boxes. I told you I would show you how E-box looks like. It's not a very selective binding sites that BMAL and clock bind to. So if you look at how many genes have this kind of binding sites, or actually they bind to tandem binding sites, you can find that 10% of the genome right there, 10% of genes have the potential for a clock-BMAL binding sites, right? So there is a very strong potential coupling just from the core clock to some of these other loops. And there are other mechanisms you can think of. If you know something about dynamical system, you know that if you have a loop, a directed loop where you have an even number of negative signs, in general, it cannot oscillate. For instance, if this concentration goes up, it will force this one to go up, will force this one to go up. So this cannot oscillate. It will go to a stable state where all the species are up or all the species are down. Whereas if you have an odd number, let's say one inhibitory connection in a loop, then if this concentration goes up, it pushes this one up, which pushes this one up, which pushes the C concentration down. So that's where you can see the potential, at least, for oscillations. You need to have in general, in dynamical system, the general rule is you need an odd number of negative interaction in order to have oscillations. So you could imagine all kinds of perturbation, changing the number of inhibitory connections in a loop, or even in loops that are connected to each other, and by this coupling, enabling oscillations that were not there, and suddenly oscillation being present, et cetera, et cetera. So you can think of a lot of coupling mechanisms. There is a lot of loops, and I think this explains why we see these widespread oscillations, which are, again, very, very flexible. I think what a cell does under a given set of genetic, epigenetic, and environmental condition is to select, reprogram itself, and select which species should be oscillating. So the set of species that are oscillating in a cell are the sort of signature, I think, of the physiological state of that cell. And now we have the methods for measuring these things. Yes, Jim?
Where maybe the majority of them are connected to these loops becomes interesting. And I know the data is incomplete, but it becomes interesting
Absolutely, I agree.
And have you looked at that?
Not really, it's the negative signature, right? So if I give you the signature of which is oscillating, you take the complement, it's gonna be what doesn't oscillate. So we're focusing on what's oscillating. It's still 10% versus 90%, so.
It would be interesting if the 10% that aren't, if you actually look at the function of those genes, if it actually made some biological sense, that they would not be on circadian rhythm.
Right, so it's actually, I mean, in a given cell is more like 90% do not oscillate and 10% oscillate. So on a given cell, the question is not, I don't think, super interesting, but aggregated, for instance. Are there things that never oscillate? Of course, that would be very interesting. And we'll do that as soon as I get back.
The other point to make about the Earth is that, as you know, computing devices generally need clocks. And in biology, you use the clocks you have available, right? I mean, we have a bunch of them. The moon, the lunar cycle, the yearly cycle, etc. So, is it possible to analyze the networks from the point of view of sort of the computation going on? To ask the question, what is the clock? And the clock is linking them all together, right? But cell by cell, can you make some inference about the nature of the computation, given that these particular ones are operating on a 24-hour cycle? The way in which you would analyze a computer by looking at the timing signal on the bus and trying to infer something about the computation that's going on in the machine based on the timing and who's tied to the timing. See what I'm saying?
Yes, I think those are very interesting and valid questions. They are not easy to study, and we're not getting direct data that impinges on that question. But I've seen, for instance, some groups that are starting to study the oscillation in the hippocampus. And it turns out that the neurons in the SCN, they are not all synchronized. They oscillate at 24 hours, but they're not synchronized, and there is actually a wave occurring. And so I think that goes exactly in your direction. So those are very interesting questions from a circadian and computational standpoint that should be investigated. Yes?
I was just wondering if there's any kind of work or anyone interested in looking at this type of, the effect of this type of circadian models at the cellular level from the computational point of view on simulation or cellular automata or something like this, to see if there's any correlation with the effectiveness of interventions of or medications depending on the circadian cycle.
So there is a little bit of work on that. I think, for instance, you could ask the question, does it matter what time of the day a given drug is given, right? And I think the answer is yes. In fact, in the Sassone-Corsi lab, they have shown to some extent that it even matters at what time you are infected in the circadian cycle, because your immune response is going to be different depending on whether you are infected during day or during night. They have done this with salmonella in mice.
But is there a way to know that? What time the infection starts?
Yeah, you can definitely control that and then measure, what's your gene expression 6 hours later, 12 hours later, et cetera, right? That's exactly, it's published in PNAS, a study on salmonella. So people are starting to do these things.
Or even for immune
yeah. Circadian rhythms are so pervasive that, any kind of biology, I think, they're going to have some kind of connection in some way.
So there is work, or there's people interested in this?
There is a little bit of work, yes. But there could be, much more could be done. I don't think nobody has studied systematically, giving a certain drugs or at, in the morning, in the afternoon, first part of the night. I haven't seen any systematic high throughput study of that. And, there is 2,000 FDA approved drugs, et cetera. But, I think the potential is there.
I have to apologize. I have a question about CRICK, but it's completely off topic to circadian rhythms.
Okay.
I noticed on the sort of the summary slide of the problem domains you applied CRICK to, there at the lower right there was EMR, and we're kind of EMR people, so we're kind of. I'm just curious if you could just briefly tell us what.
Well, that was the list of potential things. I don't work directly on EMR, We try the very small things, but we don't work directly into in EMR. But of course, if you had EMR data, you could imagine integrating with the CRICK system. For instance, the problem of side effects of drugs or drug cocktails, etc. Right? You can superimpose that data onto CRICK on CRICK networks and go back and forth.
I should probably ask other questions offline.
Okay, but it's basically 4:30. There's a lot of people I should thank, but let me just mention Sassone Corsi. This is Kristin Eckelmahan, who is the main postdoc in his lab, who has run some of these experiments. These are some of students, postdocs in my lab that have worked on motif map and circadiomics and CRICK over the years. I'm not going to go through all of them. And they look fairly happy, but I take no credit whatsoever. I try to, I'm a very mean boss, but they keep running away to the Grand Canyon and playing guitar, etc. And so that what keep them happy. So thank you very much, and I'll try to answer any questions. Yes.